Hello, good morning students. Welcome to biology class. Today's chapter molecular basis of inheritance continued and session 11 today, and in today's class I will deal about protein synthesis. But prior to speaking about protein synthesis, now let me just give some information regarding the earlier classes. Means in last class I have described about genetic code. তোমালোকৰ আগৰ ক্লাছত মই genetic code বিষয়ে ক'লোঁ আৰু তাত কি ক'লোঁ a codon is actually a part of DNA molecule, also called RNA. Kintu actually codon code directly thake to mRNA code. It is the mRNA codon that directly specify the amino acids in the protein molecule. Kintu actually codon bilak ahe obviously ultimately DNA pra ahe. But ultimately codons are born in DNA. A codon is a part of 3 nucleotide that specify an amino acid into protein molecule. Okay, codon পালো আমি codon আমি কিছুমান sense codon কলো কিছুমান termination codon কলো বা stop codon কলো sense codon actually amino acid code কৰিব পাৰে incorporate কৰিব পাৰে protein molecule কিন্তু termination codon stop codon যাক আমি nonsense codon কওঁ হিহঁতে কিন্তু protein synthesis stop কৰে গতিকে stop codonৰ function নাই কি নহয় their function is to stop the process of protein synthesis গতিকে কিন্তু before starting about what protein synthesis now let me speak about that one small part is given that relationship between base sequence and the concept of mutation. মানে mutation or এটা সৰু part ইয়াত দিয়া আছে mutation কেতিয়াবা তোমালোকক সুধিব পাৰে mutation মানে কি mutation অসমীয়াত আমি উৎপৰিৱৰ্তন বুলি কওঁ mutation actually mutation is a sudden change in the base sequence of DNA molecule as a result which there is a corresponding changes in the base sequence of mRNA take place and consequently proteins being modified. Protein modify হোৱাৰ লগে লগে characters modification হৈ যায় গতিকে he sudden change in the DNA leading into character change phenotypic expression change in the organism is known as mutation গতিকে mutation DNA ত হয় কিন্তু DNA ত হোৱাৰ কাৰণে mRNA ত হয় বা proteinত ultimately হয় okay so it is mutation you have studied in class chapter 5 principles of variation or you will find there কিন্তু এটা part one part of mutation is called point mutation actually mutation আমি দুটা ভাগত ভাগ কৰিব পাৰো mutation is two types there are two types of mutation we can distinguish one type of mutation we call chromosomal mutation one type and the second type we call as gene mutation or point mutation gene mutation also called point mutation gene mutation oblique it is also called point mutation okay chromosomal mutation involve large segments of chromosome at large segments of DNA or large segments of chromosome okay so it is gross in terms of this amount of change is very large or etu kitiba hobo pare so this chromosomal mutation can be two type again the numerical change in chromosome hobo pare chromosome number change hobo pare numerical change in chromosome or structural change in chromosome hobo pare structural change in chromosome the numerical change to kitiba ami eneko hobo paru polyploidy one type polyploidy and the other type we call as aneuploidy and the type of aneuploidy okay aneuploidy the structural change again it can be structural change a committee in dhona pawa paru four types pawa paru etu hol dilation or deficiency inversion and translocation okay egita part kintu amar ei present chapter to nohoy these parts actually belong to principles of inheritance and variation which might have been spoken by or explained by Ajanta I mean early classes গতিকে মই এটো part মোৰ ভাগত নপৰে গতিকে মই নাই কোৱা তোমালোকক কিন্তু তথাপিতো এটা লামসাম আইডিয়া এটা দি দিছো তোমালোকক আমি chromosomal mutation পাওঁ আৰু gene mutation since we are dealing with them about genes only আমি জানো gene বিলাক কি আছে genes are the sequence of bases in the DNA molecule gotike genes are very small part of the DNA present in chromosome gotike hey small part of the mane base sequence at keteba kheli meri hobo pare sometimes base sequences are altered and as a result the original character get modified jitu originally gene to asile tar jodi base sequence hogisigol he gene to gene hoin thaka beleg eta kam koribo hi gotike gene mutation lead to alteration of the phenotypic expression of organism. Hey, expression to this expression or phenotypic expression may be lethal to the organism. if the possessor dies যদি possessorটো mutation possess কৰাটো death হৈ যায় তেতিয়া lethal mutation কৰি দিওঁ কেতিয়াবা কিন্তু modification হৈ যাব alter হ'ব পাৰে sometimes it is very much dangerous কেতিয়াবা lethal হৈ যাব পাৰে গতিকে এনেকুৱা এটা good example of gene mutation is given in our book is sickle cell anemia one good example of gene mutation consequence one good example of consequence of gene mutation explained in our book is sickle cell anemia sickle cell anemia okay it holds what this is a blood related disorder relation to blood we know in our blood there are three types of blood cells present rbc wbc and platelets ইয়াৰ ভিতৰতে rbc is the main blood cells responsible for transaction or transportation of oxygen and gases and other functions in our body কিন্তু এই blood cells বিলাক normally rbc বিলাক আমি spherical আৰু dumbbell shape কাউণ্ট মানে যদি আমি dorsal view এটা দিও তেলে rbc টো দেখিবলৈ এনেকুৱা ধৰণৰ হ'ব আৰু যদি lateral view টো দিও তেলে আমি rbc টো এনেকুৱা হ'ব this is the shape it dumbbell shape lateral biconcave spherical disc kintu a shape to this shape is sometimes modified into this like a shape to enuko hoy jabe to sickle like kiteba rbc to sickle like hoy jabe pare and this is as a result of which the available space of the hemoglobin that contain inside the rbc hemoglobin containment area to komi jay and consequently ki hobo hemoglobin deficiency hobo and we suffer from anemia that anemia we call sickle cell anemia it is a genetical disorder okay and the disorder is caused by a gene located in our autosome but I mean autosomal recessive disorder boli ko to sickle cell anemia is an example of autosomal recessive disorder autosomal recessive disorder okay the gene to tamane recessive gene kintu jetia leke ei gene to dominant অৱস্থাত থাকে so long this gene that synthesizes hemoglobin actually etia ekhinte hemoglobin bikore ami kobologia a lot of information that we know that our hemoglobin is a chemically protein molecule hemoglobin is chemically a compound protein conjugate protein or chromoprotein je ko koba kintu compound protein or polymeric protein hai hemoglobin that occurs inside rbc in human being or other animals inside rbc is chemically a polymeric protein it is chemically a polymeric protein means actually it is made by 4 polypeptide chains make up it polypeptide chains make up this sidal polypeptide chain are made okay out of these 4 chains 2 chains are alpha chains and 2 chains are beta chains dudal kintu beta chain dudal alpha chain or duda beta chain pare okay itia the sickle cell anemia is an effect but it is produced due to an alteration in the beta chains of hemoglobin etu mona rakhiba beta chain or amino acid or alteration hoar karone a disease to hoy normally beta chain or in beta dural chain thake tar protital beta chain or 6th position or in the 6th position of the beta chain there is glutamic amino acid present aru it will mane 6th position of glutamic amino acid thakile detail ke glutamic acid thakiba it hobo normal hemoglobin it is normal hemoglobin and so long we no longer suffer from sickle cell anemia jetilake 6th position of glutamic amino acid thakibo kintu due to mutation in the base sequence the normal glutamic acid will be changed into valine amino acid position jodi 6th position ahi pelai 6th position valine hoar lage mon kiba due to valine some hydrophobic interaction in the hemoglobin molecule take place hydrophobic interaction in hemoglobin molecule take place and hemoglobin become conglomerated or they cluster and clog the blood vessels clogging of the blood vessels may occur ultimately because rbc which are sickle cell মানে hydrophobic interaction to karone sickle cell rbc sickle cell hai sickle cell rbc like easily cluster hobo pare ekane blood vessel to good blood to good kha jabo pare blood par hoy jabo na freely jiman freely ba smoothly jabo pare himan se jabo na gori blood transportation or badha hoy and we suffer from the disease it may be lethal sometimes hbs ami haranta ko গতিকে এটু আসলে তিয়ালে তাৰমানে বেস সিকোয়েন্সৰ লগতে যে মানুহৰ ডিজিজ হ'ব পাৰে মিউটেশনৰ আমি ৰিলেশনটা পাব পাৰো এই বিষয়লৈ আমাৰ কিতাপত ডিল কৰা আছে ওকে তো এই পথাটো আমাক দিয়া আছিলে গতিকে মই আগত তোমালোকক কৈ দিলোঁ মিউটেশন ভাৰ্চাছ DNA বেস সিকুৱেন্স এইটো পাৰ্ট আছিলে তেলে নেক্সট পাৰ্ট that we require is about the protein synthesis mechanism আমি protein synthesis processor translation কওঁ কাৰণ ট্ৰান্সক্ৰিপশ্যনৰ দ্বাৰা RNA প্ৰডিউচ হয় সেই RNA টোৰ পৰা এটা প্ৰটিন ফৰ্মেশ্যন হ'ব আৰু ফৰ্মেশ্যন অফ প্ৰটিন ফ্ৰম দ্যাট সিনথেসাইজ RNA mRNA tRNA ৰাইবোজোমাল RNA whatever be ট্ৰান্সক্ৰিপশ্যনৰ কাৰণে RNA প্ৰডিউচ হয় RNA পৰা মিউচুৱেলি mRNA প্ৰডিউচ হয় কিন্তু বাকিটা RNA প্ৰডিউচ হয় mRNA যেটি প্ৰডিউচ হয় mRNA ৰে প্ৰটিনৰ সিকুৱেন্সটো লুকাই থাকে এমিনেজ থাকিব আমি তাকে কোডন বুলি কৈছিল অলৰেডি গতিকে mRNA then ultimately leading to formation of the protein molecule by a process called translation. The translation is therefore synthesis of proteins on mRNA. Translation is therefore synthesis of proteins on mRNA molecule. আমি এইটো গম পালোঁ যে ট্ৰান্সলেশ্যন হ'বৰ কাৰণে মানে প্ৰটিন চিন্থেচিছ হ'বৰ কাৰণে কিছুমান আমাক ৰ মেটেৰিয়েলছ বা প্ৰটিন চিন্থেটিক মেচিনাৰী লাগিব তো তোমাক সুধিব পাৰে what is mean by the term protein synthetic machinery protein synthetic machinery মানে কি হ'ব পাৰে protein synthetic machinery actually it refer to the different requirements which are required for protein synthesis to perform protein synthesis হ'বৰ কাৰণে যিবিলাক requirement লাগে আমি তাক protein synthetic machinery বুলি কওঁ the protein synthetic machinery ভিতৰত major component খিনি হ'ল চোৱা the primary component is the ribosomes লাগে ribosomes are the important requirement আমি ribosomok their apparatus বা machine for protein synthesis apparatus of protein synthetic apparatus বুলি ক'লে ribosome কা বুজো okay in case of eukaryotes this ribosome is called ATS ribosome is eukaryote is ATS or in prokaryotes it is 70S ribosome in prokaryotes the ribosome is called 70S ribosome tele ATS বা 70S protein synthesis কিন্তু আমাৰ কিতাপত চোৱা মেইনলি 70S ribosome লৈ পেলাই এক্সপ্লেনেশ্যন টো দিয়া আছে তাৰমানে প্ৰকাশ্যৰ protein synthesis process দিয়া আছে গতিকে উইল ডেসক্ৰাইব দ্য process of protein synthesis with the help of 70S ribosome the ribosome টো কি হয় আমি জানো স্ট্ৰাকচাৰালি ribosome are made by two subunits the ATS ribosome is made by two subunits মানে এটা subunit আমি সৰুকে পাওঁ এটা অলপ ডাঙৰকে পাওঁ এনেকে smaller subunit of ATS ribosome is called 40S subunit or larger subunit is called 60S subunit তেনেকে prokaryote আমি এটো পাম কি smaller subunit and comparatively larger subunit smaller is called 30S subunit larger is called 50S subunit গতিকে protein synthesis 30S 50S in prokaryotes আৰু 40S 60S in eukaryotes take place okay এতিয়া রাইবোজোমস বিলাক protein synthetic machinery কওঁ এই রাইবোজোম বিলাক হয় আমি জানো they are attached to the endoplasmic reticulum and form rough endoplasmic reticulum or sometimes ribosomes may occur free in the cytoplasm যদি ফ্রি রাইবোজোম বিলাকে cytoplasmic protein ফর্ম কৰে আৰু attached রাইবোজোম বিলাক যিবিলাক ইয়াত attached হৈ থাকে সিহঁতে কিন্তু অৰ্গেনেলাৰ protein ফর্ম কৰে যিবিলাক অৰ্গেনলি ফর্ম কৰে গতিকে বেলেগ বেলেগ রাইবোজোম according to attachment they perform different functions to living cells তেলে যি নহওক one requirement protein synthetic machinery a requirement hol ribosome second requirement to hol the 20 different types of amino acids we know proteins are synthesized by the 20 amino acids they occur in sequences of different order belag sequence thakibo gotike 20 amino acids ami use kori these are the dietary amino acids mostly we require gotike all 20 amino acids they are in fact the raw materials ইহতি হল সিনথেটিক অ্যাপারাটাস ইয়াত protein synthesis হয় site of synthesis ইহত কিন্তু raw material কাৰণ protein synthesis হ'ব কাৰণে ইহতৰ incorporation হ'বই লাগিব গতিকে amino acids are the raw materials called for protein synthesis okay number third is among the requirements are different types of RNAs of different types there are three different types of RNAs required major mRNA type is mRNA to আমি সাধাৰণতে code bearing বুলি কওঁ code bearer মানে what is the role of mRNA in protein synthesis যদি আমি code bearer কওঁ কাৰণ এক মেসেঞ্জাৰ নামটো যেটো মেসেঞ্জাৰ হয় actually code bearer মানে কি carry the nucleotide sequence of nucleotide sequence of DNA from nucleus to cytoplasm it whole mRNA to function about tele it whole function second RNA is ribosomal RNA ribosomal RNA or your function to whole rule to about structural rule because ribosomes are created by this ribosomal RNA okay in case of eukaryotes ribosomal RNAs are four types কি পাওঁ এটা হল 5.8S rRNA আৰু এটা হ'ব 5.8s rRNA, এটা হ'ল 5s rRNA, 5s rRNA, এটা হ'ল 18s rRNA, 18s rRNA, আৰু আনটো হ'ল 28s rRNA, rRNA. ওকে, এই চাৰিটা কিন্তু এইকেইটা হ'ল ইউকেৰিঅটত থকা ৰাইবজমেল আৰএনএ। এইকেইটা হ'ল rRNA ইন ইউকেৰিঅটছ বা ইউকেৰিঅটিক ৰাইবজমেল RNA. আৰু ইন কেছ অফ প্ৰকাৰিঅটছ দিছ ৰাইবজমেল RNAs are 3 types. এটা হ'ল 5s rRNA, এটা হ'ল 16s rRNA, আৰু আনটো হ'ল 23s rRNA. এইটো ক'ত পাম আমি? ইন কেছ অফ প্ৰকাৰিঅটছ। এইটো মনত ৰাখিব। গতিকে যি নহওক these different RNAs perform different. তিনিবিধ আমি পালোঁ কি তেতিয়াহ'লে 3 types of RNAs required for prokaryotes and 5 types of RNAs required for eukaryotes. ইয়াত মন কৰিবা খালি মানে RNA টাইপ হৈ গ'ল কি পালোঁ আমি? ৰাইবজমেল RNA, মেছেঞ্জাৰ RNA এণ্ড ট্ৰান্সফাৰ RNA. এই তিনিটাটো লাগেই, প্ৰকাৰিঅটছত লাগে, এইকেইটা লাগে। লগতে ইউকেৰিঅটছত আৰু দুবিধ এক্সট্ৰাকৈ লাগে। এটা হ'ল স্মল চাইটোপ্লাজমিক RNA আৰু আনটো হ'ল স্মল নিউক্লিয়াৰ RNA. ওকে, তেতিয়া আমি কি ক'লোঁগৈ? দেৰ আৰ 5 types of RNAs required for formation of protein as requirements. Okay, among them আমি পালোঁ ৰাইবজমেল RNA, মেছেঞ্জাৰ RNA, ট্ৰান্সফাৰ RNA, স্মল চাইটোপ্লাজমিক RNA, স্মল নিউক্লিয়াৰ RNA. ওকে, এই 5টা টাইপ লাগিব ইউকেৰিঅটছত, কিন্তু প্ৰকাৰিঅটছত এই 3টা টাইপহে লাগিব। ইন কেছ অফ প্ৰকাৰিঅটছ ৰাইবজমেল RNA, মেছেঞ্জাৰ RNA আৰু ট্ৰান্সফাৰ RNA লাগে। এইকেইটা প্ৰকাৰিঅটছত প্ৰয়োজন নহয়। স্মল cytoplasmic or small nuclear RNA are not required in case of prokaryotes, only in eukaryotes required. So, these are the requirements, among the requirements আমি এইখিনি পালোঁ। আৰু এডিশ্যনেলি এইবিলাকৰ উপৰিও আৰু এটা ৰিকুৱাৰমেণ্ট আছিলে, নাম্বাৰ 4th ৰিকুৱাৰমেণ্টটো মই অলপ ওপৰত লিখি দিছোঁ। 4th requirement is the enzymes of different types. Number 4 to have enzymes also required in the process of protein synthesis. Enzymes to lage. Okay, among the enzymes amino acid adenylate synthetase. Okay, it is number 1 enzyme. And this enzyme to lage for activation of amino acids. Your function to ki activation of amino acids so karone ek lagibo. It is enzyme to amino acids, amino acyl adenylate synthetase. Enzyme to amino acyl adenylate synthetase required for activation of amino acid during protein synthesis. Or secondly, the next enzyme that is required, the episode enzyme to hogol ki. Next enzyme that is required during protein synthesis is peptidyl transferase. Second enzyme to likhi disu here. First enzyme to ami palu already. First enzyme to ki palu? Number 1 enzyme is amino acyl adenylate synthetase. Adenylate synthetase. First enzyme to name. Okay. Second enzyme to hold peptidyl transferase by synthetase. Peptidyl synthetase by peptidyl transferase. Synthetase or peptidyl transferase. Peptidyl transferase. Okay, this enzyme is required for formation of peptide bond during amino acid joining. Our third requirement to hold next enzyme to lagibo. To hold some of them are peptidases. Amiko peptidase. Ihon dui dhona poa jay kisuman endopeptidase. Endopeptidase সাধাৰণতে লাগে। Endopeptidase আৰু exopeptidase লাগিব পাৰে। Endopeptidase আৰু exopeptidase. Exopeptidase are 2 enzyme types that are required during protein synthesis. গতিকে এইখিনি হ'ল requirements খিনি protein synthesis. এতিয়া protein synthesis types আমি কেইটামান step পাম। So there are 4 or 5 or 6 steps that occur during protein synthesis. Protein synthesis আমি 5টা বা 6টা step পাম, বেলেগে লিখে। Gotike the 5 steps, the steps that are required during protein synthesis are as under. Steps during protein synthesis. Steps during protein synthesis. Steps during protein synthesis. Okay. Steps during protein synthesis. Number 1, number 2, number 3, number 4, number 5, and certain books written 6 steps also. First step to me call activation of amino acids. Activation of amino acids in amino acid pool. আমি এইখিনি details description পিছৰ ক্লাছত কৈ থাকিম তোমালোকক। Second step to hold acylation. acylation or charging of tRNA. Charging of tRNA, second step called charging of tRNA. Our third step is elongation of protein chain being synthesized, elongation of protein chain or polypeptide chain being synthesizing, যেটো synthesis হ'বলৈ গৈ আছে সেইটোৰ elongation হ'ব। আৰু third step, fourth step to হ'ব sorry, this is ভুল হৈছে, elongation নহয়, it is initiation of chain synthesis, initiation of protein chain being synthesized, initiation of chain synthesis. Okay, next, elongation of protein chain being synthesized, elongation of protein chain being synthesizing, and next step is termination of chain synthesis, আৰু post translational processing আমি কেতিয়া পাব পাৰোঁ? Post translational processing is a step. Translational processing also sometimes found, মানে যেতিয়া প্ৰটিন সিনথেসিস হয় তাৰ অলপ মডিফিকেশ্যন হ'ব পাৰে, ব্ৰেকডাউন হ'ব পাৰে, গোটেই প্ৰটিনটো ইনটেক্ট নাথাকিব পাৰে, তাক ভাঙি চিঙি কেতিয়াবা টুকুৰা টুকুৰ কৰি সৰু সৰু কনভাৰ্ট কৰিব পৰা হয়, আটাইখিনি পোষ্ট ট্ৰান্সলেশ্যন প্ৰচেছিং বা পোষ্ট ট্ৰান্সলেশ্যন মডিফিকেশ্যন কওঁ। Okay, tell এইখিনি হ'ল ষ্টেপখিনি, আৰু these steps I cannot describe today because they are very long to describe here. In next class I will describe. Okay, thank you.